Genome editing technologies, particularly CRISPR-Cas9, have fundamentally revolutionized the field of biology. These powerful tools offer an unprecedented ability to precisely alter the fundamental building blocks of life itself. This transformative potential, however, is intrinsically linked to a complex web of profound ethical considerations. These considerations demand careful scrutiny and ongoing, informed dialogue from all stakeholders. A primary and pressing concern in the application of genome editing technologies revolves around safety and the potential for unintended consequences. While CRISPR offers remarkable precision in gene modification, it is not without its limitations. There remains a significant risk of off-target effects, where unintended genomic regions are inadvertently modified. These unintended alterations could lead to unforeseen health problems for the individual undergoing treatment. Potential issues include the induction of cancer or the development of other novel genetic disorders. Such impacts could affect not only the treated individual but also future generations if germline editing is involved. The long-term effects of these genetic edits are largely unknown, introducing a significant degree of uncertainty. This lack of comprehensive understanding raises serious questions regarding the responsible and cautious application of these powerful technologies. Prudence dictates a thorough assessment of risks before widespread implementation. A critical ethical dimension concerning genome editing technologies pertains to the equitable distribution of their benefits. The development and application of these advanced technologies are inherently expensive, requiring significant financial investment and specialized infrastructure. This high cost raises serious concerns about who will ultimately benefit from these groundbreaking advancements. It creates a significant risk that many individuals will be left behind due to prohibitive cost barriers. Such a scenario has the potential to exacerbate existing health disparities, widening the gap between the privileged and the underserved. We face the risk of creating a situation where only the wealthy can afford access to life-altering genetic treatments. Ensuring equitable access to these potentially life-saving technologies is therefore a crucial ethical imperative. This objective aims to prevent the creation of a two-tiered healthcare system, where genetic health is determined by socioeconomic status. Policies must be developed to address these access disparities proactively. The potential for enhancement through genome editing raises profound and complex ethical questions. While these technologies hold immense promise for treating debilitating diseases, they also open the door to non-medical modifications. This distinction between therapy and enhancement is often difficult to delineate. Examples of potential enhancements include altering genes to improve intelligence, augment physical abilities, or modify aesthetic appearance. Such applications raise significant concerns about social justice and fairness within society. They challenge the very societal definition of what is considered normal or acceptable. There is a potential for new forms of discrimination based on genetic enhancements, creating a new class divide. The line between therapeutic intervention, aimed at restoring health, and enhancement, aimed at exceeding typical human capabilities, is frequently blurred. This ambiguity makes it exceptionally difficult to establish clear and universally accepted ethical boundaries. Careful consideration is required to prevent a future where genetic modifications become a prerequisite for social or economic success. Society must collectively decide where to draw the line between treating illness and pursuing genetic perfection. This discussion requires broad public engagement and expert consensus. The ethical implications of germline editing represent a particularly contentious and sensitive area within the broader discussion of genome editing. Germline editing involves making genetic changes to reproductive cells, such as sperm or eggs, or to early embryos. These modifications are then incorporated into every cell of the resulting individual. Crucially, modifying the germline introduces permanent changes that are passed down to all subsequent generations. This means that any errors, unintended consequences, or unforeseen long-term effects would become a permanent part of the human gene pool. The decision to alter the human germline carries an immense and irreversible responsibility. The potential for unintended consequences to affect future, unconsenting generations is a central ethical dilemma. This raises fundamental questions about our right to make such profound and lasting alterations to the human genetic heritage. International consensus and robust ethical frameworks are essential before considering such interventions.